Welcome to Inside the Alzheimer's Lab, brought to you by Cure Alzheimer's Fund. I'm David Shenk, author of The Forgetting and a senior advisor to Cure Alzheimer's Fund. It's a not-for-profit scientific consortium dedicated to ending Alzheimer's disease as soon as possible. We bring you this program several times a year as a series of discussions to help you get to know what the very latest is in Alzheimer's research. Today we're going to be talking with a very distinguished group of scientists. It's actually our biggest collection yet in these series of programs. Please bear with us if we run into logistical problems, but this is nearly the entire steering committee of a very ambitious new program called G2T, or Genes to Therapies, and we're going to be explaining exactly what that is in a second. And at the end of the program, we're going to be taking your questions, so please go on our Facebook page at facebook.com/curealzheimers. So what is G2T? Stands for Genes to Therapies. It's a very ambitious new program that takes the genetic discoveries that we made, which we'll be going into some detail about in a second, and actually tries to push us towards therapies, actual drugs that will slow down or stop the disease. So we're building on the successes we've had over recent years and trying to get to that all-important endpoint. And let me start by asking David Holzman from Washington University in St. Louis, David, why are we still talking about genes? We know so much about Alzheimer's on a molecular level. We know about plaques and tangles and inflammation. So why are genes still important to this conversation?
Well, I think of all the common diseases that humans get, including heart disease, cancer, diabetes, Alzheimer's disease is probably the strongest genetic disease. So while it is true, we do know that there are certain genes that were discovered now 20, 25 years ago that predispose one to getting a very early form of the disease that does link us to what we know about plaques and tangles to some extent. There's many other genes that are being uncovered now, and if you look at a playbook that's come out of cancer, where there are many different types of cancer where it's not just knowing one gene. If you know all of the different genes that are responsible for the different types of cancer, new therapies are being developed all the time that target very specific genes in each type of cancer. And I think that's really where this is going now in Alzheimer's disease, for more personalized medicine, that you really understand which genes are important in each individual person and how we can target the disease generally, but even more specifically.
So Rudy Tanzi from Harvard and Massachusetts General Hospital, you're the chair of the Research Consortium at Cure Alzheimer's Fund. You are, I think, the mastermind of G2T. Correct me if I'm wrong. So give us an overview of what this program is and how it builds on our previous efforts.
Well, over the last 10 years, Cure Alzheimer's Fund has funded the Alzheimer's Genome Project, and which I had the pleasure of chairing. And we've made quite a few discoveries in Alzheimer's genetics, along with many others in the world, in terms of what are the rest of the Alzheimer's genes beyond the ones that were discovered, as David mentioned, back in the 80s and 90s. And when we look through those genes, we're getting two broad categories: genes that directly affect the pathology that defines Alzheimer's, the plaques and the tangles, and then another set of genes that hit inflammation and the immune system. And that's the, so to speak, the three-legged stool of pathology: plaques, tangles, and inflammation. And in the beginning, we just had the genes themselves, but we didn't have the changes in those genes, the variations, the mutations that were associated directly biologically with the disease. And now, thanks to this huge effort of whole genome sequencing, where we sequenced the whole genome of over 1,500 people from Alzheimer's families, now we have the critical information we need to say, oh, here's the change in this gene that causes the disease or increases risk. To do drug discovery, you need that type of information. It's not enough just to know the gene. Genes are all good, right? You need every gene, but it's mutations in genes that will cause disease, or in some cases protect you as well. So that information is needed. And now we have it, or we're getting it. And so it's time to just kind of turn to the next page and say, let's take this information as a group and turn those genes into therapies, just like we did in the past with the first genes, learn what goes wrong from the mutations, and then come up with therapies to fix it. You can't fix something until you know it's broken.
Right.
Now we have that information. So that's it.
Terrific. Bob Vassar from Northwestern University, you get to tell us about the kind of collaboration that goes on in general with Cure Alzheimer's Fund and the Research Consortium, and how that's unique in the world of Alzheimer's research, and also how it's going to work to advance this G2T program. We see some of the key collaborators right here.
Well, what's really, I think, unique about CAF is the Research Consortium, which is a number of scientists that were brought together by Dr. Tanzi and CAF to be able to focus on the causes of Alzheimer's disease. And we have annual meetings. We just had one yesterday, and we exchange data. We work together in a very collaborative spirit to be able to try to understand the biology of Alzheimer's disease better. And as Rudy was mentioning, it's beginning to look like there are two broad categories or pathways in which Alzheimer's disease probably occurs in patients, sporadic patients. But we still have a lot to learn about this disease. The brain is the most complicated organ of the body, and we really don't understand how the brain works. And now all these genes that Rudy's effort is coming up with now suggest these two broad pathways, but we need to understand the biological function of each of these genes and how it fits into these pathways in order to really decipher and dissect the disease. That takes a lot of people. And what Genes to Therapy is all about is to target people, researchers in specific areas that are experts on those particular genes, and go to them and say, help us figure out their role in Alzheimer's disease. What are these disease gene mutations causing in terms of the biology of the gene, and how can we translate that into a therapy?
And I'd like to add that you not only exchange data, but you also exchange fairly strong views, and that's part of the
Yes.
whole, the strength of this collaboration is that you're very frank with each other at this meeting, which I was honored to attend yesterday, and it's all off the record, so you're able to especially give people inside views about the data you're coming up with as you're coming up with it, and to adjust as you're doing that. And can you give us an example? Well, I don't want to ask you to reveal something that's not public yet, but can you give us an example of how one researcher, either at the table here or not here, is going to be doing a piece of research which then will fit in with the rest of the efforts?
Well, I think the work of Steve Wagner is
Right.
these days. We saw a presentation from Steve yesterday. Very exciting news about gamma secretase, a gamma secretase modulator that he's discovered and that he's now progressing through his efforts in the preclinical phase of development, and hopefully that will move on to phase 1 later this year. I think that's a particularly exciting advance for CAF, and it's really what we're focusing on in the future in terms of gene to therapy.
Terrific. Sam Sisodia from University of Chicago. You're the mouse model guy. I think it's fair to say, among other things.
Yeah.
How do mouse models and animal models in general fit into this new G2T program?
Right. So, great question. I think that we have a lot of mouse models presently that have some behavioral impairments, that show plaques and in some cases tangles or both. And they've been extremely useful just in terms of thinking about understanding the pathobiology of the disease. Been used for testing new therapeutics, especially, for example, what Steve has been doing with the gamma secretase modulators. And it's basically, you need these mouse models as preclinical validation for your therapy before you go into humans. And what the G2T collaboration is about is to take these genes that Rudy's identified, take these genes which have mutations in them, variations, introduce them into mice using genetic engineering approaches, or delete or take the gene out from the mouse genome, and then ask what is the consequence of that of the addition of the mutation or the deletion of the gene. And what that allows you to do is then to say, gives you some information about the basic biology of the molecule in the central nervous system, where we're really concerned about it, or maybe even the periphery. We don't really know. Some of these genes may be acting in a distal place, and we know now that there's a lot of influence between, for example, the gut and the brain. And so there's information to be gleaned from the way these genes work in mice, which will eventually turn out to be, which will be important for us in the future to come to think about therapeutics that target these genes in a rational manner. So mouse models have been extremely useful in this field, certainly in all the fields of neurodegenerative diseases, and in many other conditions. But we have genetics that we use to back up what we're doing.
Right.
And that really is the strength of this effort, is that we go from genetics to therapeutics. And we can do that because when we have these mouse models, we can actually test those new models for gene action or inaction, and how that contributes to disease.
Do we have all the mouse models that we need for this program, or will we be creating?
We're going to be creating de novo all these mouse models. So
what's de novo mean? Let's explain
de novo from scratch.
Okay.
Where, as I said, there are presently a lot of mouse models out there that's being used in the community, but these are going to be brand new models because some of this data, some of the genes haven't been even published. And so we are getting, we have a very strong collaboration with a mouse genetics house called Conic, and they are building the mice for us and using very standard approaches that they're experts at, and which will be then freely used by the academic community. I think within the CAF community, and I think we'll be giving them out to other academics. So that's the idea. While
we're on this, let's take it one more step level of detail further. What does it mean to create a new mouse model? We're actually deleting certain genes from mice to create a genetically unique mouse for a certain
correct.
testing
So
process,
either
right?
deleting the gene or putting in a new gene which has, for example, the mutation, or even knocking in what we call a knock-in. You basically take this gene mutation and put it into the genomic locus of the mouse, and then say what is the consequence of that. So that's basically what this strategy is. And again, depending on what we think is going on with some of these mutations, where some of these mutations really tell you that the gene should be inactive, then we'll inactivate the gene. In some cases where the gene looks like it's still, it looks like a normal gene, nothing's really perturbed except for maybe one amino acid change, that we will have to introduce that gene mutation into the mouse.
Steve Wagner, you've already been queued up here. People have talked about your unique role in this, getting us to the actual drug. Talk about what's new with the Genes to Therapy program and how that's going to help you bring us all closer to a drug that we all badly want here.
Sure. Well, this has been a collaborative effort that brought us to where we are with respect to the first generation or second generation compounds, which were all based on these genes that David and Rudy spoke about 25 years ago. So we targeted therapeutic approaches based on what we knew about these genes which cause this early onset form of AD. Well, as Rudy mentioned, there's the majority of Alzheimer's is a late onset, right? And he's been able to, with his team, has identified a number of polymorphisms that associate with this late onset form. And so our goal is to go beyond the first and second generation therapeutic approaches which targeted these early onset genes into a more common cause of Alzheimer's. And it's going to require, as Sam mentioned, and Bob, understanding the biology of each of these genes, how these mutations actually give rise to or if they give rise to an animal model that recapitulates a lot of the Alzheimer's pathology that we see in humans. And so beyond simply understanding the biology of these genes, which is the first step, absolutely critical, what we're going to want to know is also we're going to want to understand the pathways that these genes are involved in. And that's going to require systems biologists, quantitative and systems pharmacology, because it may very well be that many of these genes aren't ideal as therapeutic targets. However, they will help us understand the pathways which enable these genes to actually cause a form of Alzheimer's disease. We'll then analyze and learn as much as we can about the biology of these genes, and that will typically unveil what I call druggable therapeutic targets. Mm-hmm. And then we'll design rationales and ideally come up with a number of small molecules that can intervene in the pathways associated with a lot of these key genes that Rudy's team have identified. So go ahead.
Well, I know you've got your hands full with your program you already have going with getting us to hopefully a drug that's very effective here. But in terms of the G2T program, are you doing a little bit of a sit and waiting game? Are there all these stages that have to happen before they get to your work, or are you actually integrated into this?
Oh no, I'm
from the get-go?
I'm very integrated. We're already doing, for example, literature searches to try to understand these pathways that these particular genes are involved in. And then what you typically want to do is to create models and predictive models where we can identify a pathway where it would be best to intervene in that particular pathway and offset the consequences of these mutations in terms of how they elicit an Alzheimer's type phenotype. And
excuse me, these models you're talking about are now computer models, not mouse models.
Right, exactly. So I think that's going to become a much bigger part of this down the road, because we were very fortunate with the first genes Rudy identified. One of them turned out to be an enzyme, and it was an enzyme that actually created amyloid deposits. And so it was very logical from a person with my background that, we can actually modulate this enzyme, prevent it from forming these lesions. And it was a very direct approach, and we were very fortunate. However, all of these genes that have come out lately are not going to be enzymes or receptors or typical drug targets. However, there's a great chance that since they show this strong association with disease, that they're going to be linked to pathways that do have targets that are very druggable that we'll identify, and then we'll start screening and do the same thing that Rudy and I have done with the gamma secretase modulators for many, many years.
Terrific, thank you.
Sure.
Tim Armour, CEO and President of Cure Alzheimer's Fund. We've had you on this program many times to talk about various functions of what Cure Alzheimer's Fund does. Let's talk about the logistics of this particular program, because you're actually on this committee because there's a lot of logistics to manage from the standpoint of a non-scientist, to make sure that the scientists have what they need, that we have the right scientists, that we have all the working parts there. So let's talk about that, and after that we'll get to the money part, which is also quite important.
Okay, thank you, David. I think one thing that's important is to thank and recognize all the people at this table, plus Dr. Wilma Wasco, who is also on the committee at Massachusetts General Hospital and Harvard Medical School in Boston, for their commitment and their guidance in this, what we think is a unique effort of putting this, all these people together, focused on a very specific objective, which is to take the genes that have been identified and understand how it is they affect the pathology, either protectively or creating more risk. Now, a lot of researchers do that individually. They share their information, but in terms of creating a plan, an organization which has a budget behind it and a timetable behind it, and going out and recruiting specific experts who already know a lot about a gene, but it might not be an Alzheimer's gene. But we've now found out through all the work that these people have been doing that it probably is an Alzheimer's gene. So we'll go to people and recruit them and say, hey, do you know that your gene is an Alzheimer's gene? And we'd love to welcome you into this effort. And to make it easier for you, this is the next thing that's, again, if not unique, certainly rare. We're going to make it easy for you by providing to you a lot of the tools that you would ordinarily have to generate yourself. So as Sam was talking about generating the mice models, we're going to do that. And when we recruit the people to work on the gene, we'll give them the mouse models. They don't have to do that. And the reagents and the support and so on. So it's a big guided effort that, I'll say again, we don't know that it's unique, but it's certainly rare in being so well programmed, having so many people working on the same kinds of things, and not just collaborating after the fact, but collaborating before the fact. So the plan is in place, the timetable is in place, we have a budget in place, and everybody's working on this together.
Let's throw out a number so we can give the lay audience a sense of just how big and ambitious this program is. Do you have a sense of what the budget will be? I know that we don't know exactly because we don't know, exactly what we're going to need here, but
I do have a sense of that
do you?
budget,
Okay,
David.
excellent.
It sort of depends on where the cutoff in the number of genes is. But if we take 50 genes, for example, and we're going to start with the first 15 of those 48, 50, 52 genes, we're probably looking at $80 to $100 million over a 2 to 3 year period. Now, Cure Alzheimer's Fund does not have all of that money. We have some money in the bank that we're going to use to jumpstart this and make sure we get a good, clean start right out of the gate. Recruit those top researchers, make sure they have the tools they need to get to work. And through the results, the early results of this, we hope to be able to encourage others to contribute and get the money and the resources we need to complete this project in a reasonable period of time. Of course, we can do one gene serially, one after the other, and be budget constrained, for the next 10 years.
Right.
And this is a classic example of where money makes a huge amount of difference. Because if we can get those resources in, we can work much more in parallel as opposed to sequence. And that also accelerates the collaboration, because you've got a lot more comparison, discussion among these people, which moves it even faster than if you have to wait for that parallel development. So that's the idea behind this whole plan. And it really takes people like this to imagine it and then implement it at the same time.
Is it fair to ask how much of that funding, what portion of it we have to go kind of full bore for, right now? Do we have 6 months worth of funding to really do it right? Do we have a year's worth of funding, or is that an unfair
we can
question?
fund the first 7 or 8 genes to get started over the next 2 years or so. And if our own fundraising holds up and increases the way that it has been and that we think it will be, we'll be able to sustain a level of 2 to 3 to 4 genes a year, something like that, which would be okay and commendable, but we really need to generate more support to take care of, again, this parallel processing that we'd like to put into place.
It's fascinating because we started out as a very ambitious organization 10 years ago. And of course, we still have the same one ambition, which is to stop this disease. But in the several years that I've been associated with Cure Alzheimer's Fund, I've seen on a day-to-day, month-to-month, term-to-term level, us getting more ambitious in terms of the number of projects we want to be funding and the kind of size of it. And this is really taking it to another level.
Sure, because the knowledge that's been gained just in the last 10 years by the people at this table as well as all the other researchers. And Cure Alzheimer's Fund in its 10-year history has funded about somewhere between 35 and 40 researchers and their laboratories and their projects. And there are thousands out there working on this issue. And the knowledge that's been generated in those 10 years has been absolutely fantastic. So not only is that knowledge fantastic and helping us to learn much more, but the science is getting bigger and the need for collaboration is bigger and the tools are more expensive. So we know more, but we also know what to do and the fact that it's going to cost more and take a lot more collaboration over time.
Let's also talk about efficiency for a second, because we're very proud that we're able to come up with ideas about things we need, or these guys are able to come up in a room like they did yesterday with new research they need, identify the researcher. We actually then reach out to these researchers and say, please send us a short, would you be willing to do this research, and we will give you the money. And it can all happen very quickly within a matter of weeks. Is that correct?
That's right. That's right. Without compromising the scientific integrity, because all those projects and the ones for genes to therapy will be very carefully reviewed, not only by this group, but our scientific advisory board, which sits at a little distance from the researchers who are actually doing the proposals. They review the proposals as well, not only for the scientific integrity, but also for the strategic importance of its contribution to Cure Alzheimer's Fund and the Genes to Therapy project.
So you have the room full of researchers that met yesterday, the research consortium, and then you've got the scientific advisory board that sits above them. And we actually had a few members also in that room, but they're essentially supervising and overseeing the work that the consortium is actually doing on the ground.
Correct. And it's important to note, I think, that unlike a lot of other organizations that have scientific advisory boards that essentially vote yes or no on a proposal, our process is much more iterative. The proposals that we get in the first place are pretty good, but if they're not quite good enough for prime time, there's iteration between the scientific advisory board and the researcher to help make that proposal better. Not just get it out the door, but how can we make this better? And that goes on a lot.
So we've been talking about this new ambition we have, Genes to Therapies, what we're hoping to do going forward. Let's actually now look backward and talk about a recent advance that we're awfully proud of that came out of, Rudy, came out of your lab in collaboration with Dr. Du Kim, I believe, and that is Alzheimer's in a dish, or Alzheimer's in a dish is the colloquial version. What is this? It got on the front page of the New York Times. Tell us what the advance was and why it helps us move forward.
Well, so for many years, we wished to have a model of Alzheimer's pathology that was complete, meaning plaques and tangles. And the amyloid hypothesis, which has been around since the mid-'80s when amyloid was discovered by George Glenner at UCSD, says that amyloid causes the rest of the disease. In other words, amyloid leads to the tangles. And we didn't have a model for that because when you put amyloid in a mouse, they don't get tangles. You'd have to do something else to them to make tangles. So some said, well, the amyloid doesn't cause the tangles. It didn't happen in the mouse. But others, like most of us, I think, at this table, said, wait a minute, the genetics says it starts with amyloid. And don't get me wrong, you can get tangles other ways. I mean, bangs to the head, concussion, insults to the brain, traumatic brain injury can get you to tangles directly. And then the tangles kill the nerve cells, and they spread through the brain. But the question was, does amyloid cause the tangles? So the main idea behind this was not just to have a very convenient model of Alzheimer's in a dish, but to ask, is it true that amyloid actually causes the tangles in Alzheimer's? And the answer was yes. So basically, when in the past, we would use just liquid cultures, what we call 2D cultures, so liquid in a petri dish.
We're looking at a slide now.
And we have a slide. And the problem is, in liquid, you can make amyloid, but it just floats away. And Dr. Du Kim, who's one of the investigators in my unit, made a very simple decision. Hey, the brain's not made of liquid. The brain's more like Jell-O, right? I mean, if you didn't have your skull, it would quiver like Jell-O. Literally, it's like gel. So the idea was, let's do this all in gel. And sure enough, when you take stem cells, human stem cells, turn those into nerve cells, cause them to make amyloid, and then have the amyloid accumulate in this gel around the nerve cells. You wait 6 to 8 weeks. You not only get amyloid plaques, that are real plaque, but you also get a tangle. And the tangle is coming from the nerve cell itself. You're not manipulating it any way to do that. So this was proof of concept that amyloid will cause tangles. And so now you have a system in a dish where you can rapidly start screening drugs 10 times faster, 10 times cheaper than in the past. And you have the confidence to say, hey, in Alzheimer's, if we stop the amyloid, we can stop those tangles, which actually kill the nerve cells and spread, as David and his group has shown, spread from nerve cell to nerve cell. So that's the idea now. And the first thing we'll do is repurpose, look for repurposing of drugs. So there are about 1,200 or so approved drugs that can be used now on this dish and say, do any of them stop amyloid? Do any of them stop the amyloid from causing the tangles? We have that part of the pathway now modeled in a dish. What's missing is the inflammation, because we also know that from the genetics that that there are certain cells in the brain. 12% of your cells in your brain are called microglial cells. And they're the little housekeepers. They help nerve cells, they clean up. They're like, I think of like the Red Cross and the National Guard when things go wrong. But if things go really wrong, microglial cells can, quickly become a SWAT team and start killing because they think there's something wrong in the brain. They think there's an infection. And at that point, they can kill nerve cells. So inflammation is a big part of the disease. We have to model that next in a new system and then merge those together. So we have two legs of the stool. We need the third leg, and we're working on that. But we're going to start screening already now. All investigational drugs or approved drugs that are ready to go into clinical trials, about 6,000 or 7,000 of them, quickly screen those for the ability to stop amyloid, stop tangles in this new model.
So I think you explained that extremely well, but there's a number of astonishing things there to a non-scientist like me and to our lay audience. Let's go back over the process there. You said we took, human stem cells, which I think also derived from just ordinary non-stem cells, right? Doesn't the process begin with non-stem cells, and we're using a technology to turn those into stem cells?
No, in this
Okay,
case,
all right,
in this case, so you can take skin cells and turn those into stem cells, and then turn those into nerve cells. In the first experiment, we just took what were commercially available human stem cells, and then we put viruses into them to up the amyloid production, and then you turn those into nerve cells. So now you have human nerve cells, and in the gel, they're forming connections like a neural network, and now they're producing amyloid. So
it's a version of a brain in a petri dish at that point.
Yeah, a very primitive brain. It's like just a nerve network in gel that we see that these nerve cells are talking to each other and making connections. And now when they produce, you've caused them to produce amyloid. And as the amyloid accumulates, because it's gel, the amyloid doesn't just float away like in a liquid. And they accumulate around the nerve cells just like they would in the brain. And then you wait, and then you see, oh, wow, now you start to see tangles, real tangles. And it's the tangles that inside the nerve cell kill. So to me, the most important thing is not just having a convenient model, but what everyone around this table has been saying for years, amyloid starts this disease. Yeah, tangles kill the nerve cells. Inflammation fans the flames, but amyloid starts it. If you want to stop this disease early, amyloid accumulates, Dave, maybe 15 years before symptoms. So hit that amyloid early and nip the disease in the bud. For patients who have it now, stop those tangles from spreading. Dave is working on that. You can stop the amyloid from being made with the gamma secretase modulators that Steve and I are working on. Bob Vassar here discovered beta secretase, the other enzyme that makes the amyloid. So there are now, based on Bob's work, there are inhibitors of that enzyme that are being tried. Much of this has been shown in animal models that Sam has produced. So we have a very special team here, I think, in terms of history of what we've done and going forward. But we need help. We need to bring in many, many more people, make this a big effort.
Let's give people a sense of what it's like to be in this room. And let me ask you, Dave Holtzman, what was your reaction when you saw the results in the Alzheimer's in a dish? Do you agree with Rudy that it basically confirms the amyloid hypothesis? If not, how far does it get us there? And so give us a sense of how you guys all work together and how you react to each other's research.
Well, yeah, no, I think it does confirm the amyloid hypothesis. I think there was a lot of information over many years of research that the accumulation of the amyloid peptide in the brain starts Alzheimer's disease. And that when this protein called tau begins to build up in nerve cells, that kind of acts as the executioner. But a lot of the evidence for that was from human beings and from animal models, but it wasn't proving it. It was suggesting what I just said, but it wasn't proving it. And I think the 3D Alzheimer's in a dish finding, to me, what Rudy said initially was really key, was just the simple idea that all of our cells make this amyloid protein all the time throughout our life, but it doesn't seem to do anything bad. It's just kind of maybe it's a waste product. We're not really sure. But when it builds up, when it accumulates and sticks together, that all the data suggested that was really started the disease. But no one, because we couldn't do that in a dish before, we couldn't test things in a way that now is much more facile. So the simple idea of just creating a gel where the cells could grow on top of each other and have material around them was brilliant, and that really allowed this amyloid to form in a dish. And once that happened, to see tau form neurofibrillary tangles after that without doing any other manipulation really sort of put the nail in the coffin and says, look, this really is what's going on. And there's a lot of data before that suggesting that's what happened, but to see that in a very simple system like this should enable more facile drug testing and also testing of other theories,
Right.
without having to spend 2 or 3 years in an animal to test something.
Great. So Steve Wagner, let's talk about the drug testing. And Rudy briefly touched on this, but the way I understand it is that it's really difficult to test drugs for Alzheimer's because you have to test them in human beings. There's a lot of ethical considerations there. There's a lot of waiting to see if the symptoms unfold. This really helps to change that dynamic, doesn't it? Because we've got a model and we can throw drugs into a petri dish, and correct me if I'm wrong, and see whether these drugs are promising enough to take to the next stage.
Yeah, David used the word facile, and really what this does, ultimately, the way you want to go about testing a compound is to first prove that pharmacokinetically it gets where it needs to go in the animal. And then after that, you want to show that it has the proper pharmacodynamic properties where you affect the biochemicals that you expect to. But ultimately, what you want to do is test it against the disease. And in this case, the disease is neuritic plaques and neurofibrillary tangles. That's the ultimate. That's what we associate Alzheimer's with, are these lesions that accumulate in brain. And being able to test that now directly in a petri dish is a huge advantage, because we used to have to rely on assumptions that, look, if we affect the levels of these particular molecules in such a way, then in theory, since these pathologies are exclusively composed of these molecules, then mathematically you should get there. But you never really know until you do these long chronic treatment studies where you can assess the effect the compounds have on the pathological process, not just the biochemical markers or the disease markers that we use along the way. So this is for guys like me that spend their days trying to identify new molecules that can interfere with this disease process, that this is a really big step forward, and it should save us a lot of time. And as we all know, time is money, and we're in a big hurry because we're in such a bad situation with this disease and the aging population. So.
Right. So Sam, I'm going to be a broken record and ask you about mouse models here, and say, do we even need them anymore? If we have Alzheimer's in a dish and all these other things,
Right.
Why do we still need the mouse models?
Well, as I said earlier, I think that it's absolutely essential that you show the efficacy of your new compound, small molecule, that while it may prevent the production of plaques and/or tangles in the dish, you still have to be able to introduce this into an animal before you go into humans.
Yeah.
And because as Steve said, the pharmacodynamics of a molecule that you get intravenously or a pill or whatever, you've got to show that this stuff gets in the brain.
Yeah.
And the blood-brain barrier is that aspect of the biology of these small compounds. They've got to traverse this blood-brain barrier, get into the brain. They have to accumulate. They have to hit the target. All those kind of things have to happen. And it's an animal, so they're not humans, but that's the best thing we have at this point. But they have to be tested in animal models before you move into the clinic.
Mm-hmm. Bob Vassar, from your standpoint, what did Alzheimer's in a dish, if it did, change your work at all? What did it add to what you can do with this inside this collaboration?
Well, it's an exciting advance. I agree with everybody's comments And what I was struck by, Rudy showed some data yesterday, that the tangles form in these human neurons in a dish by changing the ratio of two different types of tau that every neuron in the brain makes. And just by increasing the proportion of this one form of the tau, it seemed to be associated with the production of the tangles. And what's interesting too is that these are human neurons in a dish that are forming these tangles and plaques. We have mouse models where we're very, it's very straightforward to produce the plaques in a mouse model of Alzheimer's, but it's hard to get the mice to develop the tangles without introducing a separate mutation in tau that's not an Alzheimer mutation. So I think this is a big advance. I think it shows that we need both the mouse models but this in vitro model as well, where we can actually test compounds or do genetic manipulations to dissect the pathways in these human neurons in a dish, whereas in the mouse we really don't really understand how tangles are formed in the mouse. So that's, I think, is a particular important advance.
You wanted to jump in, Rick?
I just want to add that G2T includes both. So the screening for drugs in the Alzheimer's in a dish will be a big part of the G2T project and budget. And we've already begun to do that. And just to get in a little bit more detail, when we do the Alzheimer's in a dish with the gel, we can make the gel thick and then actually cut it the way you would cut up a brain and look at the pathology and screen that way. It's called high content screening. But we can also use really thin gel and then just dissolve it and look at the proteins, like how much amyloid beta protein is there, how much of the tau protein that makes tangles is there. So we're going to be doing it two different ways. And then, like Sam said, eventually if you come up with a drug, you can't get a drug into a clinical trial if you don't know gets into the brain. It has to be safe. It has the right kinetics and dynamics. So at that point, you always have to have animal models as well. But these are going to work in a complementary way. You screen quickly and more inexpensively, and with a better model, actually a complete model in the dish. Then ultimately, G2T's generating these various animal models, and then you go and validate there. So the whole thing's very integrated. We've already begun testing. We already tested a gamma secretase modulator that Steve and I have been working on for a while, and it worked really well. So that was really cool that the drug we're banking on right now, the Cure Alzheimer's Fund made possible, worked in the dish. We're testing other drugs that can hit amyloid, like hit the metal hypothesis, where we've shown that copper and zinc drives amyloid formation, and we're testing drugs that take those metals away. So we're doing it many different ways, and then the question will always be, if we stop amyloid with this drug or that drug, do we also stop the tangles? Because then you're stopping the disease process. And what's exciting is we can do this now so readily and so quickly versus the past. I think that's the real breakthrough.
Aren't we also sharing this as we come up with it? We're a nonprofit, and there are certain models out there where people are wanting to to keep their data back and to just take advantage of their own advance in what they discover. But our ethic is a little bit different in Cure Alzheimer's Fund. Once we learn something and we're content to publish it, it's out there and we want to share it with the world. Isn't that correct?
Yeah, we made the decision not to do any company or any type of proprietary thing around this system, as valuable as it is. So two ways. So if companies or people write us saying they have a drug they want to test in it, and we tell them, send us the drug, or if you want, we'll send you everything you need to do it in your own lab. We just wrote a protocol paper in a Nature journal that shows you, how to do experiments at the bench protocol. So that's going to be out there. We're shooting a video also. We're doing a video that shows you, like a YouTube video of how to do this. So we want as many people to use it as possible, and we'll send them what they need to do it. And if they don't want to do it, or they don't think they have the expertise, they can send us their drug and we'll test it, basically. That's what we're doing.
So we're going to take some questions in a minute. I want to encourage people to go on Facebook, as I said earlier, and send us some questions. We've got a couple here. Before I get into the first question, Tim, Steve mentioned the race against time. That's on everyone's mind here, but let's go over the what we're dealing with here. Some numbers in terms of how we saw this coming down the pike 15, 20 years ago. We knew Alzheimer's was going to be a bigger and bigger problem, and now it's upon us. Talk about how many people are living with this disease and what that costs the country and the world, and how that's just becoming astronomical and overwhelming.
We can start with the fact that almost half of people 85 have Alzheimer's disease. And then the next thing you realize is the baby boomer generation pushing them through that. Then the third thing is to ask, well, what does it cost to take care of that? And in 2012, the cost was about $200 billion, with a B, $200 billion.
Just in the United States.
Just in the
correct?
United States. Good clarification, yes. And most of that money was from Medicare and Medicaid. There was some private insurance money in that, but $200 billion. At the same time, the money invested by us as the public taxpayers through NIH in basic research to end the disease was about 500 million, about 400 to 1, care versus cure. And that is an unsustainable ratio. And a lot of people, including everybody at this table and their colleagues and our organization and other Alzheimer's organizations, are working with the federal government, with Congress, the administration, and others to get more funding through NIH for the basic research that really is the lifeblood of science research and affecting ends to diseases. So that's a big factor. And in fact, it's so big that $200 billion is growing every day and has the potential of bankrupting Medicare and Medicaid before the end of this decade if we don't figure out how to stop it, how to bend that curve.
And we've more than touched on that discussion, and we've given a lot more attention in other programs, and I'm sure we will in the future. So some really wonderful questions coming in over Facebook. I think the one I want to start with is, let's put this to you, Dave. We've talked a lot about genes and genetic linkage, so let's be clear about what that means for ordinary people who may think that they have it in the family or may not see it in their family. Can you explain what the actual connection is of genes to the disease? Are you guaranteed to get the disease if you have certain genes? If you don't know of anyone in your last 3 or 4 generations of your family that has the disease, does that mean you can breathe a sigh of relief and it's not coming your way? How does that work, as far as we know?
Well, there's 2 main forms of Alzheimer's disease. There's a very uncommon form that affects less than 1% of people who develop Alzheimer's disease. It's called dominantly inherited Alzheimer's disease. In that form of the disease, if you inherit a mutation or a change in one of 3 different genes called APP, PS1, or PS2, you will get the disease at a certain age, depending on the gene mutation you have and in which family you're in. So in those cases, it can be very, very predictive that you will get the disease. Now, that's not very common. There's a lot of efforts around trying to understand that early onset form of the disease by studying families who it's in and trying to look at markers for the disease before it occurs. So, and now there's 2 large trials trying to prevent or delay the onset of the dementia in families with those mutations. One is being carried out in the country of Colombia, and another one is a worldwide trial. For the most common form of Alzheimer's disease, so-called late onset Alzheimer's disease, usually occurring after the age of 60, that's a lot of what Cure Alzheimer's Disease is really working on, is figuring out what genes are responsible for that. There's one discovered many years ago called ApoE that has a big influence on risk for disease, but even if you carry the 2 forms of that gene, the bad form, the ApoE4 form, even with 2 copies, it doesn't guarantee you will get the disease. It puts you at higher risk. And many of the other genes that we're working on as part of CAF also are risk factors or they're protective factors, but they are not they don't 100% guarantee one way or another that you'll get the disease.
So Rudy, are we recommending that people do a genetic test now? I guess the answer would be yes if they suspect that they might have that first type of Alzheimer's where you actually inherit the gene and
Yeah.
you're going to get the disease. Otherwise, not
Well,
so much.
yes, you have one, like Dave was saying, you have 1% of cases that occur with onset under 50. And those are almost always genetic. Then between 50 and 60, a lot of it's genetic, so probably another 1% that would have these early onset mutations. So 1 to 2%. But for the vast majority, for these late onset cases, it wasn't enough to just know what the genes are, which is what we learned in the first phase of genome-wide studies. You need to know what's happening in the gene. What are the actual mutations, the variants in the genes that cause the disease? That's what you need to test for. Those are now just being generated because now we can sequence the whole genome. And we're compiling those. We have a database of those. Cure Alzheimer's Fund put millions of dollars into making that type of information available. We were first off the block to do it. So soon we'll be able to for the more common form, do reliable genetic testing beyond ApoE. But we're not quite there yet, and just knowing ApoE is not enough because it's not guaranteed to get the disease. But we're getting there. But for now, it's just those early onset familial forms that we can do reliable testing.
Am I right in thinking that as we get the next generation of drugs, the first generation of the drugs that actually do something about the disease, at that point we're going to want people to be tested to see how much at risk they are, or maybe which drug will correspond to their particular genome? Does that?
Well, two things. Once we have, soon we'll have enough of the genetic information to give people a good idea about their overall risk assessment. We couldn't do that before. So that's early prediction of risk. But then there's also early diagnosis. So, many of us here, especially Dave, has been involved with seeing the pathology by imaging early on, or looking in the spinal fluid for markers of the disease. So at some point, after 40 years old, everyone will do that. And just like you get a cholesterol test, or you get you check in for colonoscopy, this will become routine as we get drugs that you might need to take to stop the pathology way before there are symptoms. That's the way of the future. That's how we're going to eradicate the disease. So early prediction, early diagnosis, and then early intervention.
There's a question about how long it's going to take to come up with the drugs we need at the rate of 2 to 3 genes a year, which I think was going back to the comment you made, Tim, about what we can afford now and how we need more money to do more in parallel. Who wants to be the bold person or maybe the reckless person and say how long it's going to take, or is there any kind of model that predicts?
The model's money.
Okay.
It's the bottleneck is funding.
Right.
We're one of the most knowledge-rich, budget-constrained fields in the entire world.
So, and from a practical standpoint, can we say at all, even in terms of probability, if we moved at 2 or 3 genes a year, or 5 or 6 genes a year at this G2T in this G2T program, how long does it take to get to the point where it's likely that we will get to the right drug? Is that a fair question?
I'll let Steve comment, but I would just say that these things can happen quickly. If it's a drug that's already out there, that's approved, or one that's already gone through the first phases of clinical trials and can readily get into the next phase, it can happen quite quickly. If it's a whole new class of drug that has to be done from scratch, it could take 10 years. So it all depends. That's why you need to try everything and then hope that maybe we can repurpose a drug that's ready to go, or combinations of drugs. You do this computer-based analysis to look at where we have people we're working with that are looking at combinations of drugs, predicting whether they work together. Because even in the Alzheimer's in a dish, you can't do every combination of known drug, right? But what you can do is predict which ones might work together, and sometimes you're really surprised 2 together work, whereas each of them alone don't. Right, Steve? Oh.
Yeah, there's not really a lot I can add. I think Rudy answered the question properly. That it really just depends. To do it the standard old-fashioned way, where you start from screening hundreds of thousands of compounds, optimizing those compounds, doing all the necessary safety, tox, efficacy testing, and then doing all the clinical trials, you're talking billions of dollars, and you're talking, as Rudy mentioned, at least 10 years. But having the ability to explore a lot of other potential targets with a lot of compounds already available that have been safety tested already and been in man, it does give us a chance that we could get something quickly that that shows efficacy in modifying. We're talking about slowing the disease process. We're not talking about symptomatic treatments. We're talking about things that are going to prevent or at least slow this disease down in a hurry. So.
And Bob Vassar, I'm going to ask you the last question that came in over the transom, which is about the environment. We've been talking an awful lot about genes here. So briefly, can you give us a sense of what we know about how the environment may or may not contribute to, and the environment, of course, can be the air we breathe, the food we eat, or shouldn't eat. Let's talk about what we know on that level.
Well, that's a fascinating question. I mean, there is a portion, I think we can attribute a percentage of Alzheimer's disease to environment.
Mm-hmm.
I think we could argue about what that percentage is, but I think environment, lifestyle plays a important role in whether you are going down the road for Alzheimer's disease or whether you could protect yourself from Alzheimer's. Exercise is probably one of the best preventative sort of measures one can take. Diet, the Mediterranean diet, low in cholesterol and probably meats, I think. Mm-hmm. But also the air we breathe. We heard a really fascinating talk by Caleb Finch at USC yesterday. And he's air pollution, small particulates from fossil fuels in the air are associated with Alzheimer's disease rates. So we have a lot to learn yet about environmental factors that affect Alzheimer's disease.
A lot to learn, but let's end on that note of exercise and diet, because we do know now that what's good for your heart is good for your brain, and that includes a lot of proper eating and exercise that we've actually been advising people to do for many years. Sleep. Oh, sleep too, right? Thank you for adding that. Right. So sleep enough, exercise enough, and eat the right things, and you won't be guaranteed of avoiding this disease, but your chances will be lowered. So that is all the time we have for today. Please continue to share your thoughts on our Facebook page. Inside the Alzheimer's Lab is a production of Cure Alzheimer's Fund, a not-for-profit dedicated to ending this disease as soon as possible. Every dollar you donate to Cure Alzheimer's Fund goes directly to scientific research. That's because our board members cover all of our administrative costs. That means if you give $1, that's one complete dollar that goes directly to research. Our board members are Chairman Jeff Morby, Jackie Morby, Henry McCants, Phyllis Rappaport, Robert Greenhill, Matthew Zulik, Tim Armour, and a warm welcome to our brand new board member, Sherry Sharp. Special thanks, as Tim said earlier, to Wilma Wasco, who's also on this G2T committee and who was hugely helpful in the preparation of this program. I'm David Shenk. Thank you so much for joining us.